In this video, we are talking about the next experiment which performed to prove DNA as a genetic material. This is the experiment which actually proved DNA is a genetic material. What is this experiment? Who performed this experiment is? This is experiment 3. This is experiment 3 and this experiment is performed by Hershey Chase. Alfred Hershey's and Martha Chase. Alfred Hershey and Martha Chase actually performed this experiment and finally they proved DNA as a genetic material. Before going to know what this experiment is completely saying about final conclusions, results, everything about this experiment, first we should know about two things. These scientists used two things. The first organism they used is E. coli. These scientists used two organisms. The first organism they used is E. coli and the other organism they used is T2 phage. The second organism they used is T2 phage. We know that E. coli acts as a host for many of the experiments. Here also E. coli acts as a host. E. coli is a bacteria which is acting as a host. Whereas coming to T2 phage, it is a bacteriophage which T2 phase is nothing but it is a bacteriophage which attacks bacteria. So the name itself indicates bacteriophage. Here phage is nothing but it is a virus particle. So it is a virus which attacks bacteria. So here the bacteriophage or T2 phage is nothing but it is a particular type of virus which especially attacks the bacteria. So here they use two organisms, E. coli for host cell and the second one T2 phage. If you see the structure of T2 phage, always this T2 phage is polygonal in shape. This T2 phage is always polygonal in shape. This part which we are calling as head, this head part is made up of capsid protein. This head part which is made up of capsid protein. Extending to the head part there is a middle piece. This middle piece contains spiral protein. This middle piece contains spiral protein. And the last part of the middle piece is called end plate. This part is called end plate. Whereas attaching to the end plate there are tails. Attaching to the end plate there are tails. So these tails are generally used for adsorption on host cell. The main purpose of tails is to adsorb on the outer surface of the host. Whereas coming to the head part contains the genetic material, nothing but DNA. The head part contains the genetic material, nothing but DNA. So in the structure of T2 or in the structure of bacteriophage, we can able to identify three parts: head part, middle piece and tails. The head part contains DNA as genetic material, whereas the middle piece contains a spiral protein. And at the end plate, attaching to the end plate, there are tails. Here, this E. coli containing 2 medias. They culture the T2 phases in E. coli containing 2 medias. In the first medium which contains radioactive sulfur. The first medium contains radioactive sulfur, that is, it contains S35. The first medium contains radioactive sulfur, that is S35. In the second medium, which contains radioactive phosphorus. The second medium contains radioactive phosphorus, nothing but P32. Nothing but P32. So, in these two different mediums, they allow the bacteriophages and E. coli to grow and culture. Nothing but to multiply, they use these two mediums. If we see the first medium, that it contains sulfur. Sulfur, which is a component of proteins. Sulfur, which is a component of proteins. As we know that proteins are the building blocks of amino acids. Proteins are the building blocks of amino acids. That means here in amino acids, sulfur containing amino acids are there. In the amino acids, sulfur containing amino acids are there, like cysteine, cysteine and methionine. These three are sulfur containing amino acids. So when we inoculated or incubated the T2 phase with radioactive sulfur, what happens is the sulfur which incorporates into the capsid protein of bacteriophage. As we seen in the structure of bacteriophage, the head part is made up of capsid protein. So when we are culturing the T2 bacteriophage in a medium containing sulfur, radio-labeled sulfur, immediately the sulfur will incorporate it into the sulfur containing amino acids which are present in the protein capsid. Here in the protein capsid, all the sulfur will be incorporated. Whereas in the second medium, we have used the radio-labeled P32. We have used the radio-labeled P32. We know that phosphorus is not present in the proteins, but it is present in DNA. Phosphorus which is present in the DNA. Phosphorus which is a backbone of DNA. That means now the radio-labeled P32 will bind it to the DNA which is present in the T2 bacteriophage. So here the sulfur will bind to the protein part, capsid protein part, whereas radio-labeled P32 will bind to the DNA which is present in the T2 bacteriophage. So when this bacteriophage allowed to infect bacteria, that time what happens is this is the E. coli bacteria. So E. coli bacteria is having its own chromosomal DNA and plasmid part. So what we are doing here, we are inoculating or we are culturing the E. coli bacteria along with the T2 bacteriophage. So now the T2 bacteriophage which is radio-labeled with sulfur. So here the T2 bacteriophage is radio-labeled with sulfur. That means the sulfur has been incorporated in the head part, protein part. That means here this part will incorporated with will incorporate with sulfur. When they allowed to culture in E. coli, they occurs in 4 stages. The first portion is adsorption. Adsorption is nothing but sitting of T2 bacteriophage on the host cell. Here, this T2 bacteriophage has been attached to the outer surface of the E. coli bacterium. So, the first step is adsorption, attachment to the outer surface of bacteria. And the second part is injection. Here is nothing but piercing. So this is the bacterium. Now this T2 bacteriophage perforates or pierces the host cell here, and it allows its DNA to enter into the host cell. Now this is the T2 bacteriophage DNA. The genetic material which is present in the capsid part now allowed into the E. coli bacterium. So this is the bacterial chromosomal DNA. This is an extra chromosomal DNA or extra genetic element, nothing but plasmid. Whereas coming to here, this particular DNA is viral DNA which got incorporated by ingestion process. So when this sample has been centrifuged, centrifugation, it rotates at a high speed. During this high speed, what happens is this protein part which is lesser in weight, whereas the bacterium which is heavier. So whereas the bacterium which is heavier, always the heavier object settles at the bottom. Here also the heavier object here, the heavier organism here is bacterium. When we centrifuge it, the bacterium settles at the bottom, whereas the capsid is left at the top. Nothing but supernatant. The bacterium settles at the bottom, whereas the capsid left as supernatant. Supernatant contains capsid protein which is radiolabeled, whereas the palate contains bacterium. Fine. So here in the supernatant the capsid protein which is radiolabeled. So finally after the centrifugation process, the radioactivity is observed in supernatant. Finally, after the centrifugation process, the radiolabeling or the radioactive nature is observed in the supernatant. That means in the protein part, the supernatant contains head part, nothing but capsid protein. So the radiolabelability is observed in the supernatant. Whereas in the second medium, when the bacterium and T2 bacteriophage is grown, same E. coli is having its own bacterial chromosome apart from the extra chromosomal DNA. Now the T2 bacteriophage sits on the outer surface of bacterium. That is nothing but this is adsorption process. That means the T2 bacteriophage So once the replication completes, it undergoes a process called lysis. It kills the bacteria, or it pierces the bacteria, and other DNA, the duplicated DNA will release it out. The duplicated DNA will be released out in the last step, that is in the lysis. After this process, again same like the previous, when they get centrifuged, the heavier object got settled as a pallet. The heavier bacteria settled as a pallet, and the lighter object, nothing but capsid protein, left as a supernatant. So at the pallet we can able to identify the radio label. Why? Because in the pallet this genome is identified in the bacterium. In the second medium, what happened? The phosphorus, which is not present in the protein, but the phosphorus which will definitely present in the DNA. So now here the DNA have been incorporated here. That means in the pellet, if you see in the bacterium, the DNA, radio labeled DNA has been identified, whereas the supernatant which we can see is a capsid protein which is non-radio labeled. If you see the first centrifuge tube and second centrifuge tube, in the first centrifuge tube we have seen the radio labeled protein. In the second centrifuge tube, if you see, we can able to identify the radio labeled phosphorus 32, nothing but bacteria. That means here the protein is not responsible for transformation, or protein is not responsible for all the changes here, but DNA is responsible for transformation. So from this experiment they come into conclusion that DNA acts as a genetic material.